Good afternoon. My name is Davide Camoni from Enoconsulting. I'm honored to be the first speaker of this module. We will talk about light struck taste. I promised I would not mention chemical formulas in order to be clearer while talking about this defect. In my module, specifically, we identify possible sensory descriptors, thermodynamic and kinetic models behind this defect of wine, and then we will also draw conclusions about possible ways to counter this defect. First of all, gusto di luce in Italian is a translation in that it is not a taste issue. Gusto stands for taste, but it is a nose problem, an odor issue, due to a number of elements and chemical compounds which basically give a defect in terms of odors, and I'm talking about DMS, DMDS, mercaptans. Riboflavin is very active in presence of proteins which have sulfur lateral chains, methionine specifically. And therefore, there is an acceleration of this production. There is another aspect. Riboflavin is an optical active element. It means that it reacts with light in a wavelength which could be brilliant yellow from 380 or so nanometers to 400. When this light struck effect is present, there is a side effect, which is the reduction in the brilliant color of the wine. And if there is a prolonged effect, we have an increase in the browning of the wine. There is a third series of phenomena which is not linked to sensory precursors, but which occurs nevertheless, and it does in wine. It is iron oxidation. This is something that has been mentioned already today. You have glyoxylate, hydrogen peroxide, a number of substances which have an influence on oxidation of wine. So please bear this in mind, because oftentimes people think that the light-struck taste is a phenomenon per se, which simply has an impact on the odor, but that is not the case. Photooxidative reactions are of different kinds. Reactions of type 1 include all those reactions that include and involve chemical bindings. In reactions of type 2, we have all reactions that involve the participation of individual radicals. Then we have collateral side reactions on carotenoids and fatty acids. Carotenoids degrade and acrolein are formed in the presence of light. The result is what is known. Normally, carotenoids are weak protectors of acids, and as a matter of fact, they permit the oxidation of cinnamic acid. This is something that was already mentioned before in relation to the formation of oxygen, also by way of this channel. There are rapid activation systems in the lab. The methods are simple. You have transparent see-through bottles. They are activated by light sources. Just to give you an idea, a bottle on the desk or bench of a laboratory or on a shelf develops this defect in 1 month. In control conditions, a number of tests have been performed with red, green, blue LEDs, and in that case, you have the defect within 8 hours. we used very specific LEDs that work on the wavelength of riboflavin, which is 390 nanometers. And in that case, the phenomenon basically took just 1 hour to be completed. What you see there are also direct and indirect variables. The light-struck taste phenomenon occurs more rapidly when the bottle is transparent, when the glass is clear, when the filtering is diminished, the filtering effect is diminished, and when the ratio between the exposed surface and the content of wine is low. These are variables that you can, of course, also experience. There are then indirect variables such as the SO2 content, the alcohol content, and temperature. Their impact is not as immediately clear because you have to make further analysis. With reference to temperature, consider that temperature does not accelerate this reaction. A bottle in the fridge under a neon light at 5 degrees has the same kinetics when it comes to light-struck taste than a bottle out of the fridge. Above 30 degrees, and we know that based on Australian studies, there are other synergetic factors that come into play. So one first element to be considered here is that a lowering in temperature, in this case, cannot increase the shelf life of the product. This is the Australian work and study. They investigated transparency to light of the various bottles. It is a famous work. You see here that between 300 and 400 nanometers, de facto, see-through and bluish clear bottles have very high transparency, while darker bottles are not totally non-transparent. This is our lab, 6 labs, increasingly clear and transparent. The first one is dark, covered in aluminium and wrapped in aluminium. Then the second one is clearly dark, and I don't know whether you can appreciate on the slide the transparency. But here we have variable transparency plastic films, and the dark bottle, as a matter of fact, is not protected either. The defect is bigger in sample number 6. Here you have the percentage and the amount of riboflavin, but the defect was present also in the dark bottles. So the light-struck defect occurs in dark bottles as well. It is just a question of time or transparency to light, and longer exposure times can bring this defect to light. There are antioxidant substances to be considered. Here I mentioned ascorbic acid, but I could also focus on polyphenols. There's a wonderful work about ascorbic acid from the 80s when ascorbic acid was analyzed in relation of co-degrading riboflavin so that it does not react with methionine, with tryptophan, and with other compounds which can cause odor defects. 32 co-degradation chemical pathways have been identified with riboflavin affinities very high. Here you see that in 180 minutes of light, riboflavin and ascorbic acid degrade by 4 logarithmic orders, which means that more than 90% of compounds disappear. So there are ways that can be adopted in order to avoid this reaction. Having said that, I would like possibly to explain or to present to you 3 theoretical models. We tend to think that oxidation is one way, but as a matter of fact, there are also Renew's logics to be considered, meaning by lowering temperature you have a longer shelf life, and that is one case. But in other chemical reactions you have the Nernst equation coming into play, so that wine includes antioxidants and it is a sort of battery. So it is the relationship between that and precursors that establish whether a redox is stable or not. And photoelectric oxidation is what is considered in the Planck-Einstein system. The problem, as I said before, is the fact that iron and tartrate form oxygen and glyoxylates, and this leads to a number of chemical reactions. I will show a number of them to you. You see here sulfides, sulfates, and as a matter of fact, the result is that shelf life diminishes, and this is what counts for us. So riboflavin is an element which is B2 vitamin, and it is a component of wine. There can be reactions in the presence of light, and you might need to minimize that if you decide to go for transparent, clear glass bottles. But please remember that also dark glass bottles are subjected to this defect. There are ways to intervene, and the next speakers will focus on that. There are still studies that need to be performed. A couple of trials were made in 2012. So there is still a long way to go in terms of analyzing things. That what is important is to know that we can combat this phenomenon. Thank you.